Good day, everyone. I am Maureen Lauzon, and today I will discuss my assigned topic, the lungfishes. In this presentation, there are 5 parts. I will first discuss the lungfish's taxonomy or lineage, then its distinguishing group traits, its global representatives, their distribution, and lastly, their adaptive structures and mechanisms. For the first part of this presentation, we will discuss about lungfish taxonomy. The lungfish are sarcopterygian air-breathing fishes that belong to the subclass Dipnoi. Dipnoi actually means double breather due to the presence of both gills and lungs. Lungfishes have been around since the early Devonian epoch, which is roughly 420 million years ago, and most of the orders and families have been wiped out due to extinction. Nonetheless, there are 2 extant orders, Order Ceratodontiformes and Order Lepidosireniformes. Order Ceratodontiformes has 1 extant family, family Neoceratodontidae, which also holds 1 extant genus, the Neoceratodus. Meanwhile, Order Lepidosireniformes holds 2 families, Lepidosirenidae and Prototheriidae. The 2 families have 1 extant genus each, which are Lepidosiren and Prototherus respectively. Collectively, these fishes are commonly termed as lungfishes. However, there is no local name for lungfishes here in the Philippines. Now, we move on to the distinguishing group traits of lungfishes. First, we discuss their visible characters. Lungfishes are characterized by their lobe-paired pectoral and pelvic fins, one of the key features that distinguish them from the ray-finned fishes. Instead of having rays as support for their fins, they have bones. Second visible character is that their caudal fin combines with the dorsal and anal fins. Third character is that they have plate-like teeth. And lastly, they have gill slits or external gills despite having functional lungs. We will discuss more about their lungs later in the presentation. Figure 1 shows the visible characters of a gilled lungfish. Now let us discuss their ecology. For their salinity, fossil records suggest that these fishes were also found in the marine environment. However, the current species are strictly confined in freshwater pools. Their diet consists of insects, crustaceans, worms, mollusks, small frogs, small fish, and any plants that they can come across, making them voracious omnivores. The way that they feed is that they first suck in the food, then crush and chew it using their teeth. No stomach assists in their digestion as they only have a spiral valve. Lastly, for reproduction, all extant lungfishes are oviparous and show no sexual dimorphism except in Lepidosiren, which I will discuss later. However, there are still differences in the way that they breed. Lungfishes of the genera Prototherus and Lepidosiren spawn during the start of the rainy season. They also provide parental care by creating nests for eggs and by provision of protection by the males. Oppositely, the Neoceratodus do not provide parental care and they only lay their gelatinous eggs on water plants. For the last distinguishing group trait, let us talk about how lungfishes share the most similarities with tetrapods than any of the other fish groups. Some studies on phylogenetics suggest that the lungfishes are the closest living relatives of tetrapods, while some believe that these fishes undergone convergent evolution, making this a debatable topic. Nonetheless, the similarities to tetrapods are undeniable. The overlapping characteristics of lungfishes and tetrapods include homologous lungs, which permit pulmonary circulation, skull bones in fossil records, enamel in teeth, four limbs in the same arrangement as tetrapod legs, brain and reproductive system, which is very similar to amphibians, as well as the ciliated skin in embryos, just like in amphibians, and many more. Moving on, let us now discuss the extant representatives of lungfishes. Unfortunately, there are no Philippine representatives for these fishes. Therefore, I will discuss all 6 global representatives alive today. First of the 6 is the Neoceratodus forsteri, or the Australian lungfish. It is the only extant member that belongs to family Neoceratodontidae of the order Ceratodontiformes. It is the most primitive surviving member of the ancient air-breathing lungfish. Also, it is only found in Australia. These lungfishes set them apart from the others because they retain the primitive structures of ancient lungfishes. These structures are flipper-like pectoral and pelvic fins, and also that they are heavily ossified. Their larvae also differs from the others because of the internal gills present instead of the external ones as found on the others. They breathe only with one lung and rely more on the gills. They only breathe atmospheric air when stressed or active. Lastly, they can survive a number of days out of water. However, it is only possible if the environment is moist. Nonetheless, they cannot withstand total water depletion, which makes sense as to why they cannot estivate. I will discuss more about estivation later. Next up is Lepidosiren paradoxa, or the South American lungfish, as shown in Figure 3. It belongs to family Lepidosirenidae of the order Lepidosireniformes, wherein it is the only existing member alive today. This fish is isolated in South America, wherein they are found in the swamps and slow-moving waters of the Amazon, Paraguay, and lower Parana River basins. In contrast to the Australian lungfish, they own a pair of lungs. Also, they need to breathe atmospheric air because their gills are greatly reduced and almost futile in adults. However, when mating season comes, they breathe through their gills when breeding. Males also show or grow additional but temporary branches on their appendages. These branches supply additional oxygen to the eggs in their nest. This is also a manifestation of their sexual dimorphism. Lastly, they undergo estivation, but they do not release mucus. Last for the global representatives are the African lungfishes. These fishes belong to family Protopteridae of the order Lepidosireniformes. Currently, there are 4 species of African lungfish present today. They are usually found in swamps, backwaters, and small rivers in West and South Africa. During their larval stage, they have external gills that facilitate gas exchange. Nonetheless, like their South American counterparts, they also have a pair of lungs and are obligate air breathers when become adults. Similarly, they also estivate in times of drought. The only difference is that they secrete mucus while the South American lungfish does not. Now, here are the 4 African lungfishes, namely, first, the Protopterus doloi, or the spotted lungfish. found in Figure 5. They are usually found in the river basins of Congo and Gabon. Second is the Prototherus aethiopicus, or the marble lungfish in Figure 6. They are found in the eastern and central Africa, as well as the Nile region. They are the largest of the African lungfishes and are also known to have the largest genome of any vertebrate. Third species is the Prototherus amphibius, or the gilled lungfish in Figure 7. They are found in East Africa, and they are known as the smallest extant lungfish in the world. Fourth, and lastly, the Prototherus annectens, or the West African lungfish in Figure 8. They are found in West Africa, and this lungfish has a prominent snout and small eyes with cycled scales embedded in the skin. We are now on the fourth part of this report, the spatial distribution of lungfishes. We will first discuss its historical expansion. The oldest recorded fossil of lungfish, which is Diabolicleus, belonged to the early Devonian period. Research showed that their diversity thrived during the later part of the Devonian time. However, it gradually declined during the Triassic period. Although diversity momentarily revived during that time, it nonetheless declined once again. The historical distribution of lungfishes was first explained by Vicarians. If we view it from the evidence of Vicarians, it would suggest that the primitive distribution of lungfishes were confined only to the supercontinent Gondwana of the Mesozoic era. However, fossil records present a more historical background for their distribution. Fossil records show that the advanced lungfish had a wider freshwater distribution, wherein the current distribution of lungfish species showed the extinction of numerous lineages due to the separation of Pangaea, Gondwana, and Laurasia. Furthermore, Cretaceous fossils show that some lungfishes were also traveling to marine waters by having uri-haline qualities. Here are all the recorded occurrences of lungfish. As shown in Figure 9, lungfish show a cosmopolitan distribution and also a recorded occurrence in the marine environment. The extinct Ceratodus of the order Ceratodontiformes was believed to be found all over the world. On the other hand, here are the recorded occurrences of lungfish over the last 20 years, according to the GBIF network. These records will show us where extant lungfishes are probably present or most likely to occur. The Neoceratodus species inhabit Mary and Burnett rivers of Queensland, Australia. Meanwhile, the Lepidosiren lungfish lives in the tributaries of the Amazon. Lastly, Prototherus species are restricted to the fresh waters of Central Africa. Finally, we are down to the last segment of this presentation. We will now discuss the adaptive structures and mechanisms of lungfishes. First adaptive structure is their long, stringy pectoral and pelvic fins. In all lungfishes, except for the Australian representative, have long, stringy appendages that are in constant motion. The tips of these fins have a highly developed sense of touch. This adaptation aids the almost blind lungfish. Touching and sensing the environment using their tips makes up for the low vision. Next, we will discuss the ability of lungfishes to perform electroreception. Aside from their highly sensitive tips, lungfish also have electroreception. What is electroreception? It is the ability to detect electrostatic fields in the environment. This mechanism is very helpful for lungfishes in detecting prey as well as communication. Moreover, lungfishes possess a lateral line on the body or on their head and/or electroreception sensors in the snout to detect these electric fields. Next, we talk about the lungfish lungs and how they breathe atmospheric air. Unlike other fishes, the lungfish possess a highly specialized respiratory system. Derived from swim bladders, their lungs enable them to breathe in atmospheric air. Despite not owning a trachea, the pharynx and larynx are connected to the lungs. Respiration is also much more optimized due to the presence of numerous honeycomb-like air sacs that increases surface area for gas exchange. This is shown in Figure 13. As presented by the Australian lungfish, they also breathe differently from other fishes, wherein they breathe through their nostrils with their mouth closed. This is made possible because they have a choana or choana, which is an opening that links the nasal cavity to the mouth. This structure is also present in tetrapods and not in ray-finned fishes. As for their breeding techniques, they actually relocate to water holes during times of drought. In these water holes, they solely breathe atmospheric air to survive. Also, in lungfishes, the oxygen-rich blood and deoxygenated blood do not mix. This is because pulmonary circulation and systemic circulation are partially segregated by a valve in the truncus arteriosus of the heart. Figure 14 shows a schematic of circulatory system of the South American lungfish. Last, but definitely not the least of adaptive structures and mechanisms of lungfishes is estivation. I will now discuss its importance and how it is done. So what is estivation? Estivation is defined as the state of dormancy or dry sleeping of lungfishes as a way to combat desiccation when dry conditions occur. Only African lungfishes and the South American lungfish perform this behavior. In addition, African lungfishes can estivate for 4 years max. Meanwhile, the Australian lungfish cannot estivate since it heavily relies on water or moisture to breathe. As shown in Figure 15, the lungfish starts estivation by digging and burrowing in the almost dry mud. So you may ask, what happens during estivation? When finally positioned into the mud, African lungfishes ensheath themselves using mucus to trap moisture. This prevents them from desiccating. Meanwhile, the South American lungfish does not release mucus. They only estivate bare. So what happens to their metabolism when under estivation? When under estivation, the body temperature of the lungfish drops significantly. Its metabolic rate also drops as little as 1/60th of the normal rate. So how do they obtain nutrients if they're just sleeping? Lack of nutrients is not an issue for the sleeping lungfish, as it eats away its own muscle. Lastly, how do they deal with protein waste? They deal with protein waste by converting it into urea. Urea is a less toxic form of nitrogen waste as compared to ammonia. That is it. That is all about lungfish. I hope you learned something from this presentation. Thank you all for listening, and I hope that you have a great day today. Please stay safe and God bless you all.